Hello and welcome to my channel Omni Bioentrance, and today we will be discussing next 2023 Shift 2 question paper. I will be discussing only the biology section. If you are new to my channel, please do like, share and subscribe. Coming to the first question. An example of multiple alleles is seen at the locus that determines the feather pattern of mallard ducks. One allele M produces the wild-type mallard pattern. A second allele MR produces a different pattern called restricted, and the third allele small md produces pattern termed as dusky. In this allelic series, the dominance pattern is capital MR greater than capital M greater than M small d. In a cross between restricted and mallard ducks, it was found that only dusky ducks were absent in the F1 generation. This indicates that the genotypes of the parents most likely could be. So I have done the crossing, the various types of crosses what they have mentioned here. I have done all of them in the next slide. So restricted and mallard, and this is 1 minute. And this is dusky phenotype. Small md, that is dusky phenotype. Now you have to see for which cross will give you the dusky phenotype, will not give the dusky phenotype. So capital M, capital R, capital M into capital M, small m d. Now only small m d occurs only once here. Only if in both the parents small m d is present, then you will get the dusky phenotype. See over here the crosses. MRM, MRMD, MM, MMD. So there is no dusky phenotype upon crossing these two parents. So these two parents could be one of them. Then coming to the next parent combination, MR, MD into MM. So if you cross here also, M small d occurs only in one of the parent, not seen in both the parents, so no dusky phenotype. This is the cross. Now if you see option 1, it is MRM into MMD. Here also no dusky phenotype. But the other combination what they have mentioned, you will get the dusky phenotype. So option 1 cannot be the answer. Then in option 2 also, second combination has MD genotype in both the parents. So it will produce dusky phenotype. And option 4 also not correct. They have not given all the combinations in option 4. So the correct answer will be option 3. Then next one. UPGMA is a method of constructing phylogenetic tree using distance matrices between organisms. The following matrix depicts distance measured as the difference in characters between 5 organisms. The distance between a pair of organisms, say P and Q, and the third organism R is calculated as an average of their individual distances from the third organism. Example, new average distance between PQ and R is R to PQ. R to PQ. So PQ here will be 60, then Q will be 50 divided by 2, so it will be 55. Now I have done how to construct the phylogenetic tree using UPGMA method using distance matrices between organisms. I have done in the next slide. Now based on the distance matrix, the correct phylogenetic tree is. This is what they have asked in the question. We will construct the phylogenetic tree using the data given. So here is the first data that they have given. I have taken it and pasted it again once more over here. So first what you have to do is identify organism with the fewest difference between them. So QP and TS, or we can say PQ and ST also. That is 20. So draw the first grouping of a phylogenetic tree. That is over here. If you see here, this is the first grouping of a phylogenetic tree, PQ and ST. Now next what you have to do is determine the average distance which PQ show to others. Now PQ to PQ, no answer. Then it is 0. Then PQ to R. So that will be PQ to R. PQ to R. $60 plus 50 divided by 2, that will be 55. Then PQ to S. So PQ to S, that is first P to S, that is 100 plus P to Q to S, that is 90. So 100 plus 90 divided by 2, you will get 95. Then PQ to T. So first it will be P to T. So that will be 90. And next will be Q to T. Q to T is 80. So 90 plus 80 divided by 2, you will get it as 20. Now next what you have to do is proceed to ST grouping. Now you have done for PQ grouping. Now you have to do for the ST grouping. Make a table as done above. So this is the PQ ST grouping table. So PQ to R we already know from the previous table, it is 55. Then PQ to ST. PQ to ST will be PQ to S. From the previous table it is 95. And I have not taken the previous table in this diagram. So I'm extremely sorry for that, but I will tell you the values. PQ to S is 95 from the previous table, and PQ to T is 85. So 95 plus 85 divided by 2, you will get it around 90. Then R to ST, R to ST, R to ST over here. So it will be R to S, it will be 40, and R to ST to and R to T, not ST, R to T, that will be 50. So 40 plus 50 divided by 2 will be 45. Now what you have to do, the next step, identify the organism with the fewest differences between them. So here as per this table it is 45 which is having the fewest difference, and that is ST to R. So you have to draw a grouping like this. From ST draw a line, it will be 45. Now then next you have to construct a STR table which is also done as previously. So PQ STR, PQ STR. Now PQ to STR, that will be PQ to ST, that is PQ to ST is 90, and PQ to R, PQ to R is 55. So 90 plus 55 divided by 2 will be 72.5. That you have to draw as shown in the options, that is like this. So from R to PQ. That is 72.5. So the answer here will be option 1. It's better if you do it on your own and see, then you check whatever I have told. Then you will understand the concept easily. It is very easy actually. Then next one. In the given pedigree, circles represent females and squares represent males. Filled shapes indicate affected individuals while unfilled shapes indicate unaffected individual. Based on the pedigree information provided below, identify the inheritance pattern. This is the same kind of pedigree but done in a different way for autosomal recessive condition, which we have discussed in the previous shift 1 paper. So for autosomal condition, I can tell you that even if the parents are not affected, the child is affected. and if male is affected, male and female children are affected. So it has nothing to do with X-linked dominant and X-linked recessive condition. Then you can see there is skipping of generation. Even if these individuals are not affected, but their son is affected. So there is skipping of generation, both males and females are affected, and even if parents are not affected, children will be affected. So that is the conditions for autosomal recessive. So the answer here is option 2. Then next one. Restriction enzyme recognize certain sequences within the DNA and cleave them. If a DNA fragment is cleaved with BAMH1 restriction enzyme, it generates sticky ends. If the same DNA fragment is cleaved with SAV3A restriction enzyme, it generates sticky ends. The cleaved fragments can be joined using DNA ligase. The recognition and cleavage site for BAMH1 and SAV3 are given below. N represent any of the nucleotides. So this is for BAMH1 cleavage site and this is SAV3A cleavage site. So based on this information and assuming there is only a single cleavage site, choose the correct option. So these are the options and this is my working out for these options. If SAV3A cleaved end is ligated to a BAMH1 cleaved end. So this is BAMH1 cleaving, this is SAV3A cleaving, and these are the cleavage products for BAMH1 and these are the cleavage products for SAV3A. Now if what they have told, if SAV3A cleaved end is ligated to BAMH1 cleaved end. Now this is BAMH1 cleaved end, this is SAV3A cleaved end. Both of them are ligated, you will get the product like this. You can even ligate these two also, I have shown only one. So this is the ligation product that you get. The ligated fragment can be further digested using SAV3A irrespective of the neighboring sequence. Now SAV3A, this is N. N means it can be any nucleotide. So it can be G over here, no problem, SAV3 will digest. Here also it can be 2N are there, it can be any of the nucleotides, SAV3 will digest. So first option is correct. Then next one. If a BAMH1 and cleaved end is ligated to a SAV3 cleaved end. Same way, if BAMH1 is ligated to a SAV3 cleaved end, that is suppose these two are ligated. The ligated fragment can be further digested using BAMH1 irrespective of the neighboring sequence. Now these are the ligated product of SAV3A and BAMH1 cleaved fragments. Now here it is N, it is not G. So BAMH1 is not going to cleave that. So second option is incorrect. Because N has to be always G for it to be cleaved with BAMH1 and not any nucleotide. Then third option. If both the recognition sequences are reverse complemented, then it cannot be cleaved using either BAMH1 or SAV3. Now this is the BAMH1 sequence. Now reverse complement is, this is the complement of this sequence. 5 prime 3 prime 3 prime 5 prime. So 3 prime 5 prime is the complement for this 5 prime 3 prime sequence. Now reverse complement means just read it in the 5 prime 3 prime direction. That is why I have drawn the arrow like this. So you have to read it from GGATCC. That is the reverse complement of BAMH1. This is the reverse complement for SAV3. Now what they have told, if both the recognition sequence are reverse complemented, then it cannot be cleaved using either BAMH1 or SAV3. Now if you read this reverse complement, and we know that the restriction enzyme 2 sequences are palindromic, you can cleave them with BAMH1 and SAV3 respectively if they are reverse complemented. This sequence is same as this sequence. So it could be cleaved with BAM H1 and this one could be cleaved with SAU3A. So third option is also incorrect. Then fourth option. If BAM H1 cleaved end is ligated to a SAU3A cleaved end and reverse complemented, then the ligated fragment can be digested using BAM H1 irrespective of the neighboring sequence. Now this is the reverse complement and BAM H1 cleaved end is ligated to SAU3A. We already did that one previous slide. And if you take the reverse complement of that, you are going to get an N over here. Now BAM H1 is not very happy with the N over here. It wants C itself in its position to get it cleaved. If not C, it is N, so it will not be cleaved. So option 4 is incorrect. Then next one. A mutant bacterial strain having a shorter glycolytic pathway was discovered. If the mutant bacteria are grown aerobically, the net ATP yield was lowered to 28 compared to the net ATP yield of 34 from Krebs cycle for wild-type bacteria. Except for the reaction that is bypassed in the mutant, assume that the other reactions of the pathway remain unaffected. The step that is most likely bypassed is. So you have got a mutant bacteria and it has got a shorter glycolytic pathway and it yields a 28 ATP molecules compared to 34 from Krebs cycle for wild type bacteria. So which reaction was affected? Which pathway was affected? Now there is a loss of 6 ATP molecules, 34 minus 28. And these 6 ATP molecules, they come from the step when glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate by the dehydrogenase enzyme. Here 2 molecules of NADH are produced which will yield 6 ATP molecules. Because 1 NADH, it will yield 3 ATP molecules. 2 NADH, it will yield 6 ATP molecules. So in the first option, phosphoenol to pyruvate, 2 ATPs are generated. Fructose 6-phosphate to fructose 1,6-bisphosphate, ATP is broken down. 1,3-bisphosphoglycerate to 3-phosphoglycerate, ATP is generated. So the correct answer is option 2. Then next one. The table below represents the kinetic data obtained for an enzyme in the absence and presence of 2 different inhibitors P and Q, each at a concentration of 10 millimolar. Consider the total enzyme concentration is the same for all experimental conditions. P and Q respectively are. So they have asked for what type of inhibitor is P and what type of inhibitor is Q. Now what I have done is I have drawn a graph for easy finding out the answer. So here you have got substrate. This is without inhibitor. This is with inhibitor P and with inhibitor Q. These are the values they have given. So first I have to draw a graph for the without inhibitor. That is 1 by S, 1 by V0 values they have given. You can draw the Lean-Weaver-Burke plot. So 1 by S, 1 by V0. This is the graph for 1 by S, 1 by V0. This is only a rough sketch based on the values. These values that has been given. This is only a rough sketch. Now the next graph is for 1 by S, 1 by V0 in case of inhibitor P. This is the line for inhibitor P. Now we can see that it is an uncompetitive inhibitor. Why? Because in uncompetitive inhibitor, the inhibitor binds only to the enzyme substrate complex rather than the free enzyme. It does this by binding to an allosteric site which is different from the active site. The end result of this is that Vmax is lowered. That is why the graph is over here, not on this line nor on this line. It is on a different line, that is over here. It is having a different value which is lesser than that without inhibitor. So Vmax is lowered as well as Km is also lowered. Km for inhibitor is over here and for without inhibitor it is here. So Km is also lowered. So it is a case of typical uncompetitive inhibition. Now for Q I have done in the next slide. So this is for Q. So this line, it is for without inhibitor, and this line is for with inhibitor. 1 by V, 1 by S values are taken. This is the Km value. These are the Vmax value. Now it is from the graph, we know that it is a non-competitive inhibition. So what happens there is, in non-competitive inhibition, the inhibitor binds to both the free enzyme and the enzyme substrate complex with equal affinity. So in such cases, what happens? Vmax decreases but Km will remain the same. So Km is same over here, but Vmax is decreasing. So the answer here will be, first one will be P will be uncompetitive inhibition and Q will be non-competitive inhibition. So option 3 is correct. Then next one. Clutch size in birds refers to the number of eggs laid in a single nesting attempt by a nesting pair of birds, while number in brood refers to the number of young hatched. The graph below represents the relationship between these parameters, that is clutch size and number in brood, and fitness in bird populations of the great tit. The y-axis for samples 1 and 2 for graphs 1 and 2 could respectively be. Now if you see the options, you will see that average weight of young, average adult survival, average net dimension, all these do not fit to the clutch size graph, because clutch size refers to the number of eggs laid in a single nesting attempt by nesting pair of birds. So how can you know adult survival and weight of the young? It is not possible. Even average nest dimension also not possible. So it should be average number of clutches. So graph 2 should be average number of clutches, means average weight of young for graph 1 is suitable. So the answer here will be option 4. Then next one. A population has 3 genotypes XX, XY, YY, where X is dominant over the Y allele. The number of each genotype in the population is as follows. XX 1185, XY 3045, YY 1300 individual. Consider that there is a random mating, no gene flow, no mutation and selection, and the population size is sufficiently large. The correct statement is. Now we have to apply the Hardy-Weinberg principles for this case, because they have told that there is random mating, no gene flow, no mutation, no selection, and the population size is sufficiently large. So the correct statement is, we have to see that I have done in the next slide. So what they have given here is genotype XX, XY, YY, and if you add all these things, you will get the total as 5530. That is observed number, not frequency they have given. They have given the observed number, that is 1185, 3045, 1300, and total adding all these you will get 5530. Now from this value you have to calculate observed frequency. So observed frequency will be 1185 divided by 5530. It will be 0.21. Similarly for XY and YY also calculate and you will get these values. Now if you add up this value and this value, it is coming up to 1. So observed frequency is in Hardy-Weinberg equilibrium. Now we have to see the Hardy-Weinberg expected frequency. For that we have to calculate the P and Q values. P of X I have written here, it is actually small p. So that is calculated using XX frequency plus half of XY, because half of it is contributed by the X allele. So it will be 0.485. Similarly Q value, that is PY value, same formula, but only thing, instead of XX you will be putting YY. So 0.24 plus 0.275 you will get it as 0.515. Now the Hardy-Weinberg expected frequency, X square or P square. So what you will get? 0.235. How? Multiply 0.485 into 0.485. You will get it P square. So it will come up to 0.235. Next is 2pq. We have to find the value of q first, q square, or we have the value q over here. That is 0.515 into 0.485 into 2, you will get 0.4995. And q square, if you see, it is 0.265. Upon multiplying 0.515 and 0.515. If you add up all these values, that is p square plus 2pq plus q square, you should get 1 if it is in Hardy-Weinberg equilibrium. But here you are not getting it as equal to 1, so it is not in Hardy-Weinberg equilibrium. But it could become Hardy-Weinberg in equilibrium if it is there is random mating allowed for one generation. So that is your answer in the options. Then coming to the next question. The life cycle of yeast Saccharomyces cerevisiae, which reproduces both sexually as well as asexually, is depicted below. M and OP represent. Now M represent germination, N represent vegetative growth of the haploid cells, O represent vegetative growth of diploid cell, and P represents starvation. All the other options are incorrect. Only option 1 is correct. This I have drawn it colorfully in the next slide with explanations. So this is it. These are mating between haploid cells of the opposite mating type, A and alpha. These are the diploid cells after mating, A, alpha. Then there is vegetative growth of the diploid cells, that is by budding. Then during starvation causes ascus formation and result in meiosis, resulting in 4 haploid ascospores contained within the ascus. Then the ascus ruptures and the spores germinate. Then vegetative growth of haploid cells. Meiosis has already taken place here, so it will be haploid cells. These are the haploid cells, and you will get the products after the vegetative growth. Then these will again become diploid cells, then budding, then again starvation period, ascus formation, and the cycle continues. So the option here, M is for germination, N is vegetative growth of haploid cells, O is vegetative growth of diploid cells, and P is starvation. So option 1 is correct. Then next one. The population sizes of two organisms P and Q growing in a given habitat is shown. If P and Q share ecological relationship, then they most likely represent. Now P, if you see over here, it starts with a higher population than Q. That is its predator. So P is the prey. But in option 1, they have given P as predator, so that is not correct, and Q as prey, so that is not correct. So the correct option will be P will be herbivore and Q will be carnivore. So the correct option will be option 3. Then next one. Female beetles prefer males with bigger mandibles. In an experiment, a population of these beetles were picked and divided into 2 groups. For one group, only those males who had larger than average mandible size were allowed to mate to produce next generation offspring, that is group 1. So in group 1, the males have larger mandibles, and for the other group, the males and females were allowed to mate randomly. So it can be larger mandibles or smaller mandibles, so it is random mating. That is in group 2. These populations were maintained using this regime for 50 generations. After this, it was found that the male mandible size in group 1 was significantly larger than that of group 2. However, the females in group 1 produced fewer offsprings than females in group 2. Possible explanation for this observation is, so in the experiment, as selection on female reproduction was not imposed in group 1, female reproductive capability declined over time. That is not correct. Group 1 males produced offsprings with larger thorax to support larger mandible and hence smaller abdomen, which influence the egg carrying capacity in female offspring, leading to a decline in female reproductive ability. That could be one reason. So option 2 is correct. Under limited food condition, males with larger mandibles in group 1 preferred females with lesser reproductive ability, as that allowed dominant individual male to have more resources for themselves. That is also not correct. Under limited food condition, female producing fitter offsprings after mating with males with larger mandibles started producing fewer offsprings to nourish them better. That would be more correct. So option 2 and option 4 are correct. Then next one. Autoradiography of a green leaf of summer squash, that is this upper panel. This panel showed import of C14 carbon from the source over a period of time. A similar experiment was carried out with an albino tobacco leaf, that is this. Albino means it doesn't have chloroplast. So it will not photosynthesize. Shaded portions denote C14 labeling. These shaded portions, they represent C14 labeling. Based on these observations, the correct options are. Now, before coming to that explanation, let me tell you one thing. Plants produce carbohydrates, that is sugars in the leaves by photosynthesis. But non-photosynthetic parts of the plant also require carbohydrate. For this reason, what they do is they will translocate their carbohydrates from sources. That is, sources means the regions where excess carbohydrate is present. Now that is seen in mature leaf, not in young leaf. Now young leaves, they act as sinks. And what are sinks? Regions where carbohydrate is needed more. That includes your root, flowers, fruits, stems and developing young leaves. These are the sinks. So carbohydrate has to be transported from source to sink. Now source usually is your mature leaf. So in the source, carbohydrate labeling will be lesser. C14 labeling will be lesser. That is why the mature leaf is having less shaded portion when compared to younger leaf. Similarly over here also, even though it is albino, they are checking only for the transport of sugar. Now based on these observations, the correct option is, in the early stages of development, the leaf act as a source. I already told, in early stages of development, leaf act as the sink. So option 1 is not correct. Mature leaf gains the ability to load and export sugar. That is correct. That is why from here it is being transported to the younger leaves. So here it is less C14 shaded portion is present when compared to higher C14 in or more shading in younger leaf. The import to export transition is dependent on the developmental stage of the leaf irrespective of photosynthesis. Yes, because in albino leaf also, the younger leaf is getting more, is showing more C14 labeling than the mature leaf. So import to export transition, it is dependent on the developmental stage of the leaf irrespective of photosynthesis. This is smaller leaf and this is bigger leaf. Similarly, this is young leaf and this is mature leaf. Next one, import cessation and export initiation are two separate events. That is also correct. Now options 2, 3 and 4 are correct here. Now next one. In a true breeding homozygous lines of snapdragon antirrhinum majus, white colored flowers of personate shape was crossed with red colored flower with pleoric shape. The F1 were pink and personate shaped. Assuming that both these genes segregate independently, choose the correct options. So these are the options that they have given. I have done it in the next slide. So this is the first option. F2 progeny will have 1 by 4th probability of showing the parental phenotype. Now they have crossed white, that is capital W, and red is small w. Personate is capital P and pleoric is small p. Because they have told that in the F1 generation they have got personate, so personate must be the dominant allele. Now when you cross capital W, homozygous varieties, that is WW capital P to small w, small p, these are the gametes that are produced. This is the F1 progeny. So capital W, small w, they already told in the question that the F1 progeny is pink and personate. So capital W, small w must be pink in color, and capital P represent personate character. Now they have asked for the F2 progeny. Now F2 progeny is obtained by crossing the F1, self-pollinating F1. So these are the F1 gametes, these are the F1 parents, and these are the F1 gametes. All these are F1 gametes. Now you have to do the Punnett square over here like this, and you have to name the phenotype of each offspring. So what they have told, F2 progeny will have 1 by 4th probability of showing the parental phenotype. Now if you see the parental phenotype, what is the parental phenotype? Pink personate. So if you see for pink personate, you can see that it is over here 1, 2, 3, 4, 5 and 6. So 6 out of 16 will be showing parental phenotype. So there must be a printing mistake as per me. I am not sure about it, because if it is parental phenotype, then it will be 6 out of 16. And if it is parental genotype that they have asked, then you will get 1, 2, 3 and 4. So 4 out of 16, that is 1 by 4. So it will show 1 by probability of showing the parental genotype. Maybe I am wrong also. If you have any thing to tell about this answer, please let me know it in the comment section. Then next option, 50% of the progeny in the F2 generation will be pink in color. Now which all are pink? 1, 2, 3, 4, 5, 6, 7, 8, 8 out of 16. So it will be half. Half means 50%. So 50% of the progeny is pink in color. So that is also correct. In F2 progeny, pleoric shaped flowers with pink color are expected to be in 1 by 8 ratio. So pleoric shaped and pink color. Pink pleoric, that is this one. 1, 2. Yeah, 2 of them are pink pleoric. So it will be 2 out of 16. So it will be 1 by 8. So in this progeny, pleoric shaped flowers with pink color are expected to be 1 by 8 ratio. Then next question. So next question, what they have told, specific fluorescence probes are used to label proteins present on the surface of specific immune cell type. A scientist labeled protein P with green probe and protein Q with red probe. A machine can provide quantitative information about the amount of these two proteins present on the surface of each cell by quantifying 1000 cells. This experiment is repeated for cells present in the blood of multiple individuals who are healthy young, healthy aged with cancer and with autoimmune disorder. The data of 10 individuals per group is provided below. If there are no other confounding factors, then based on this data, the correct inferences is or are. First one, in healthy aged individuals, that is over here, the expression of P reduces drastically as compared to Q. It doesn't reduce drastically as compared to Q. It is 10 raised to 2 over here, and it is somewhere around 10 raised to 3 over here. So it doesn't decrease drastically. Then reduction of Q protein can be correlated with the development of cancer. That is over here. This is cancer and there is reduction of Q protein. It is 10 raised to 1 whereas P protein is more, 10 raised to 5. So option 2 is correct. Increased expression of Q protein can be correlated with autoimmune disorder. This is the Q protein 10 raised to 5. There is increase whereas P is very much reduced. So it is autoimmune disorder. So that is also correct. Option 2 is also correct. In comparison to healthy individuals, that is these, the expression of Q in autoimmune condition is negatively regulated by expression of P. That is also not correct. So the correct options here are 1 and 2. The next one. The following graphs depict three different scenarios where the average body size of a population, assuming a normal distribution with a single mean of a study organism, has been plotted over several generations. If body size is heritable and there is no genetic drift present in the population, the correct options that can give rise to the observed patterns would be. Now this is average size in population and these are the generation P, Q and R. Now P they have given as directional selection, so this is here selection is active. Natural selection types they have asked. So directional selection, it occurs when environmental pressures favor one phenotype more than the other. As you can see in this graph, only one phenotype is favored more than the other as in P. So P is showing directional selection. That is correct. Then disruptive selection, a mode of natural selection in which extreme values of the trait are favored over intermediate values. So extreme values of the trait are favored over here than the intermediate values. So R is showing disruptive selection. Then Q is showing either no selection or a stabilizing selection, that is stabilizing selection in which genetic diversity decreases as the population stabilizes on a particular trait value. So it can be no selection or a stabilizing selection. So option 1 and 2 are correct. So that's it. Thank you for watching my channel. Please do like, share and subscribe.